Hello, my name is Mr. Baker. This is the third lesson in my fifth unit for my biology class. We're going to continue talking about modern genetics. We're going to specifically look at how gene therapy is used to treat different pathologies. And we are, again, going to kind of be just skimming the surface here. This is some very cutting edge science, and it's going to be changing all the time. And it's also very in depth. It's very easy to get lost in the deep end if we go too in depth. So we're just going to make sure we hit the high points here and talk about some of the general things to know about gene therapy and using it to treat sicknesses, diseases, illnesses. All right. So genetic engineering, changing a genome, right? So we already talked about genetic engineering. So that's rewriting or inserting or changing the nucleic acid bases in DNA to change a trait about an individual, and we usually do that in vitro, right? So genetic engineering to change a genome in order to treat an illness or a disease. So for treating these things, they must be due to a genetic mutation. If it's something that you catch, we can't fix it with gene therapy. We're not going to fix the common cold with gene therapy right now. You know, I'm not a futurist, so I'm not going to predict if that will happen or not. But I will say that if it is a genetic mutation that causes a disease, chances are likely, especially if it's due to a recessive disorder versus a dominant disorder, that gene therapy may be able to be applied in order to treat that illness. Right? So why is it better for the illness to be recessive? That's because if it's recessive, the individual only has one allele for that illness. Because remember, recessive means it's in a heterozygote or a homozygote recessive. And if they're in homozygote recessive, that means we can insert a single dominant allele, and the dominant allele will be expressed over the recessive allele. Now that means that if we're going to do this kind of gene therapy, if we do it in a somatic cell, a body cell, it's not going to be as effective as germline cells. Germline cells don't affect the individual, though. They affect their offspring. So this would be if you have a disease or an illness that is genetic and it's going to be passed down to your kids, and you want to not pass that down to your kids, gene therapy could be used to treat the illness in vitro before fertilization happens, or in a zygote, which would be pre-fetus right after conception. Right? However, germline gene therapy is illegal in multiple countries. And we're talking science here, not talking ethics, so I'm not going to go into why it's illegal. Just want you to know that it is illegal in multiple countries. All right, so this is very similar to the genetic engineering we just talked about, where we're going to take the new DNA that has been modified or engineered, right? And we're going to put it inside usually a virus to make recombinant DNA and viral DNA, as this poster says, right? And that virus can actually infect the cell. So it's actually going to be let into the cell past that cell membrane. And in this image here, it's being wrapped in a vesicle like most larger things are when they enter the atmosphere, enter the cell. And the virus itself breaks that vesicle and then is able to insert itself through the nuclear pore and inject DNA into the nucleus. Right? This is just an example. It's not how every single thing happens, but this is from the National Institute of Health here in the U.S. So I thought it'd be good to kind of go over as an example of how gene therapy is done. Right? There are some issues with gene therapy besides the ethical that some folks might have. Usually gene therapy takes multiple treatments, right? Which means that the individual has to undergo multiple not necessarily surgeries, but injections or exposures to the recombinant DNA. And because it's recombinant DNA, their body might have an immune response to it. So their body might actually fight it off. Or they'd get very sick because of the viral infection that's being used to insert the DNA into their cells. Right? So the viral vectors can have some danger to certain individuals. If it's a multi-gene disorder, all this gets amplified because they have to go through multiple treatments for each gene that can trigger the illness or disease. And if there are there may be problems with inserting the DNA into the cells where their cell is either able to fight it off, or once it gets into the cell and into the nucleus, the new DNA may be fixed by those things we talked about way back when we talked about mitosis and meiosis. When DNA is replicated, there is a error checking that goes on involved with that. So it may get picked up by an error checking enzyme. I almost said protein. Let's use a little bit more specificity in our words, though. And then this gene therapy is expensive. It costs a lot of money to do this. And so if you're undergoing this multiple times and you're doing it multiple times, especially for a multi-gene disorder, this adds up and it's very, very costly. Right? So right now, it's not a good answer to those illnesses and diseases, but sometimes it's our only answer. All right, so we've talked a little bit about how gene therapy is used to treat different pathologies. If you're watching this for my environmental science class, we're going to spend the next few days going over this a little bit more in detail and looking at how gene therapy is actually used to treat specific pathologies. So, as always, keep learning and keep asking questions. Have a good one.